The structure and function of blood vessels. The main blood vessels are arteries, veins, and capillaries. Arteries. Arteries are blood vessels that originate from the heart and carry blood away from the heart. Arteries are deep-seated in the muscles. The aorta carries oxygenated blood from the heart to all parts of the body except the lungs, while the pulmonary artery carries deoxygenated blood to the lungs. Arteries have a thick muscular wall and a narrow lumen. The thick wall is to resist the high pressure of the blood inside the arteries. The artery wall has 3 layers. The innermost layer of the artery is the endothelium. It is composed of a single layer of cells. This layer is found in all blood vessels. It provides a smooth lining which offers the least possible frictional resistance to blood flow. The middle layer is composed of elastic fibers and smooth muscles. The outer layer is made up of inelastic collagen fibers. When ventricles contract, the muscular layer of arteries relaxes, stretching outwards to allow blood into the arteries. When ventricles relax, the muscular layer contracts, pressing inwards, forcing the blood to flow forward. This produces the pulsating action in arteries. The muscular wall of the arteries is under the control of nerves and hormones that alter the diameter of the arteries. This plays a major role in regulating blood flow to the body organs. The ability of the arteries to alter the diameter of their lumen plays an important role in regulating blood flow to the body organs. The aorta is the main artery that leaves the heart. It forms branches, which supply blood to major body organs. These branches divide to form small blood vessels known as arterioles. The arterioles branch further to form blood capillaries. These capillaries ramify the body tissues. Veins. Veins are blood vessels which carry blood from all parts of the body back to the heart. Veins tend to lie close to the body surface. The walls of the veins are thin with less elastic fibers. Their lumen is relatively wider compared to arteries. This offers minimum resistance to blood flow. Blood pressure in the veins is very low due to the immense resistance to the flow at the capillary bed. To prevent backflow of blood and ensure that blood flows only towards the heart, veins have valves throughout their length. In addition to the valves, forward flow of blood in veins is maintained by contraction of skeletal muscles. Large veins, for example in the legs and arms, are located in between the skeletal muscles. When the muscles contract, they press the veins, forcing the blood to flow towards the heart. This explains the need for physical exercises. All veins carry deoxygenated blood except the pulmonary vein, which carries oxygenated blood from the lungs to the heart. Generally, blood in veins contains high levels of metabolic wastes and low levels of nutrients. Exceptions to these are the renal vein and the hepatic portal vein. Blood in the renal vein has low level of nitrogenous wastes, as these will have been removed in the kidney. The hepatic portal vein carries blood rich in nutrients that have been absorbed at the gut. Capillaries. Capillaries form a very important part of the circulatory system. They are numerous and very close to the tissues in such a way that each cell is near a blood capillary. Exchange of substances between the tissues and the blood takes place across the capillary wall. This is possible due to the fact that the wall of capillaries is made up of the endothelium, which is only one cell thick. This provides the least distance for exchange of substances between the capillary and the cells. The capillary walls are permeable to water and molecules of small size in solution. Branching of arterioles into capillaries increases the surface area of the capillaries and pressure of blood in them. The pumping force from the heart together with the narrow lumens of the capillaries exert a high pressure that forces the fluid part of the blood to filter out of the capillary walls into the intercellular space, forming tissue fluid. This occurs by ultrafiltration, also known as pressure filtration. Tissue fluid is a watery liquid that is similar to plasma in composition, except that it lacks proteins, red blood cells, and some white blood cells. Phagocytic white blood cells are able to squeeze in between the cells of the capillary wall into the tissue fluid. The tissue fluid bathes cells of the tissues and forms a medium for exchange of materials between blood and the cells. From the tissue fluid, cells extract substances such as glucose, oxygen, amino acids, vitamins, hormones, and mineral ions. Into the tissue fluid, the cells release metabolic waste products, e.g., carbon dioxide and urea. Ultrafiltration results in a drop in pressure such that at the venule end of the capillary bed, the blood pressure is lower than that of the tissue fluid. The proteins retained in the capillaries during formation of tissue fluid exert a high osmotic pressure at the venule end of the capillary. This causes much of the tissue fluid to be drawn back into the bloodstream by osmosis. Other substances such as carbon dioxide diffuse across the capillary wall into the blood. The remaining tissue fluid drains into the blindly ending lymph vessels and joins the lymphatic system to form lymph. The lymph eventually joins the bloodstream through the subclavian vein located in the neck region. The venules from the capillary bed join up to form larger vessels called veins. Differences between arteries and veins. The walls of arteries have thick, muscular, and more elastic fibers. The walls of veins are less muscular and have fewer elastic fibers. Arteries have no valves except at the base of large arteries leaving the heart. Veins have valves at intervals throughout their length. Arteries have a narrow lumen. Veins have a wider lumen. Arteries carry oxygenated blood except pulmonary artery. Veins carry deoxygenated blood except pulmonary vein. Blood flow in arteries is in high pressure. Blood flow in veins is in low pressure. Blood in arteries flows in pulses. Blood in veins flows smoothly with no pulses. Arteries have ability to alter diameter of the lumen. Veins have no ability to alter diameter of the lumen. Thank you for watching this biology lesson. Please subscribe to this channel and other Genius channels relevant to your level of study.